We're covering the layers of the gastrointestinal tract, and we start with the small intestine. Most texts and books will talk about the small intestine first, but then we'll break down as we get through the esophagus, the stomach, the small intestine, and the large intestine that it's made up of 4 layers: mucosa, submucosa, muscularis or muscularis externa, and serosa. And you'll see tunic or tunica referring to layer. There's 4 layers. Students won't find this very exciting because you're looking at a lot of slides for this, but we're focusing on these 4 layers that's continuous throughout the gastrointestinal tract. Parts of the layers have different structures because we're changing the function of the organ. But as we look at these layers, mucosa is inside or most superficial where the food is touching, and then serosa is the outside. Serosa is the outside of the tube, and we call that visceral peritoneum early on in the curriculum. The only difference is when we look at the esophagus and the rectum, the outside's called tunica adventitia because it's a different function. 4 layers. So we are looking at a model to show you where the food would pass and then the outside of the tube. Mucosa, referring that there's mucus produced throughout the gastrointestinal tract, that's this layer. And in the small intestine, the food is running along all of these hills and valleys that we call a villi or villus for singular. Along these villi that we can't see on this model are microvilli. Microvilli help with absorption. Remember, the small intestine, its primary goal is to absorb nutrients. So as we pass food along this is 1/2 of the tube. So if you took this model and flipped it, it creates the whole pipe. And what do I mean by that? If we just look at this histology real quick, we are looking at one section of this here, this square. So the food is running through here, but we're just taking one piece of this model to start. As I look at this, where the food touches, mucosa. Within mucosa, there's 3 types of tissue: epithelial, connective, and muscle. So just understand mucosa to start is very important. When we look at the lining of these villi, it's lined with epithelial tissue, specifically simple columnar epithelium. So just this outside lining, where it's pink all throughout, this is simple columnar epithelium, and this again is where the food is touching. It changes color and it turns white, or dark white or stained white. This is all now my lamina propria. The lamina propria is connective tissue. This is loose connective tissue. And you notice this trend continues throughout the body. When you learn integumentary system, you have epithelial tissue, then connective tissue underneath or deep to it. Same thing. So epithelial, simple columnar epithelium is the lining of mucosa. Deep to it, lamina propria, that's my loose connective tissue. And then I have this thin strip of muscle that we call the muscularis mucosae. This is smooth muscle. This is the end of tunica mucosa, or that first superficial layer. This can be very confusing. So mucosa is 3 layers of tissue. The superficial, simple columnar epithelium. Then I have connective tissue, which is this lamina propria underneath, and then lastly this smooth muscle tissue, which is muscularis mucosae. It is a smooth muscle. Remember, you can't control it, but it's helping to create that wavy action for tunica mucosa to contribute to the absorption and then also secreting some of the juices throughout the small intestine. That is the end of mucosa. Below it is submucosa. I tend to have some glands and blood supply in submucosa. And then I get into muscularis or muscularis externa because I have more smooth muscle to help move the food along. So we use peristalsis and segmentation. And I keep saying food, but at this point it's now being digested. So we have an inner circular layer and then an outer longitudinal layer. So throughout the digestive tract, you have 2 layers for this muscularis: inner circular and an outer longitudinal. The only difference is for the stomach, it has 3 layers. It'll have an oblique layer, then a circular, then a longitudinal. Remember, the stomach has a lot of mechanical mashing of the food to start, so it has that extra layer of muscle. So this is my 3rd overall layer is muscularis, but it's made up of 2 regions. Circular, because it's wrapping the tube in a circle, and then longitudinal because it's running the length of the pipe. So they complement each other. Lastly, serosa is the outside of the tube. Remember, we called that visceral peritoneum. This is serosa, referring it's secreting. Remember that membrane that helps secrete, but it's the outer layer. So 4 layers: mucosa, submucosa, muscularis, and then serosa. The hard part's about mucosa. It's 3 types of tissue: epithelial, connective, and muscle. And then we get into muscularis. You have 2 layers, circular and longitudinal. So let's take this model and expand it into the histology of this. And different slides are different, but really what I'm doing is taking this box and expanding this segment here. The food is touching along the villus or villi. So all of these in my mind, and some of this is broken in this slide, but let's look at these are fingers. When I see fingers, to me that's the villus. So each one of these fingers is one villus, so they contribute to the villi. What sits on top of them are my microvilli for that absorption. Remember, when we look at mucosa, we're just looking at the first part that's got 3 types of tissue. So when we go back from our model and extrapolate it to this, the lining of my villus or my villi is simple columnar epithelium. When I go inside where I have the fingers, the meat of the finger, this is my lamina propria. The way I think about it again is the skin is my simple columnar epithelium. The meat inside is my lamina propria. So the lining of a villus is simple columnar epithelium. Inside is loose connective tissue that we call that the lamina propria. I have goblet cells in there to secrete mucus. That's why it's called tunica mucosa. I also have other intestinal glands or duodenal glands as well, because it's specific to the duodenum. And those are those glands as we get down to the base. The last piece of mucosa is our 3rd type of tissue, smooth muscle. That's this strip of muscle that runs across here. This is Muscularis mucosae. Some slides look different, and that's why it's important to just remember the order. Mucosa, submucosa, muscularis, and then serosa. If you remember the order, all I do is look for changes. So I know I have my villi, I know I have my lamina propria, and then I hit these glands, these little bubbles here, but then I see this strip that runs across to support that absorption. That's my muscularis mucosae, and it's got a pinkish appearance to it, or peach. But this thin muscle, muscularis mucosae, I know that's the end of mucosa. So everything underneath here is all submucosa. So all of this is submucosa, where I'll have blood supplied to support the organs because I have connective tissue underneath. And then I get into a change where it turns bright pink. Well, this is muscularis. I have an inner circular and an outer longitudinal. The circular tends to be a wider muscle for the small intestine. So all of this is circular, circular, circular. And then I see a change as this runs across. This is longitudinal. So 2 muscles for muscularis when it comes to the small intestine. Circular, longitudinal, and then the very outside capsule, just that dark lining, is tunica serosa. So as you go through your histology, understand first mucosa, that it's simple columnar epithelium, then my connective tissue, which is lamina propria, then my smooth muscle, which is muscularis mucosae. All of that includes mucosa. Then I can get into the easier parts of submucosa, muscularis, which is circular, longitudinal, and then the outside of serosa. Remember, this will be continuous. There's a couple changes here and there from esophagus all the way through large intestine, but the 4 layers stay the same. So all you're doing is repeating some of this as you go through histology. We just pick out the different structures that are unique to each organ. So this is small intestine histology.